Tim, thanks for coming back, and let's say welcome to Tim.
Well, thank you, John, for all those complimentary things. I'm very happy to be back. This is an update on the talk you would have heard last time. So let's go viral. Don't need to really say anything about that. And that's to remind you what the name of the virus is. Who is going to say COVID-19? No, it is SARS-CoV-2. And the SARS bit stands for Severe Acute Respiratory Syndrome. There was another SARS, but that was back in 2002, and it only lasted for a year in, guess where, China. So, do you realise that COVID-19 is not a virus, which is the name of the disease caused by the virus? Here we have a little picture of the likely origin of SARS-CoV-2. The primary host is thought to be in little bats that are about that big in wingspan. The average bat carries 12 types of viruses, but it's not affected, so it's literally a carrier. And it carries the viruses, but they don't give it any symptoms. But it then goes and bites civet cats or raccoon dogs. That's the latest one. And then the Chinese, bless them, go to their wet market and they say, oh, I'll have a leg or a side of civet cat for my tea. And then it's transferred to Homo sapiens that way.
Excuse me, if you're away from the microphone.
Oh, you can't hear, okay, yeah.
Thank you.
Thank you. So let's now check your understanding. We're going to be talking about transmission of the virus or shedding of the virus. So I'm going to ask you two questions. Does COVID-19 attach itself to water droplets and then travel in aerosols from person to person?
Yes.
Keep asking yourself, why do we wear masks? So there we are, that was my first surprise. It is not COVID-19 that attaches itself, it is SARS-CoV-2 that attaches itself. And surprise, surprise, can you shed COVID-19 in a sneeze or a cough? So you shouldn't get this one wrong.
That's probably no.
So I found that clever little diagram of a virus wearing a mask. Okay, the best available picture to the general public, is that sort of thing which you can find on ABC websites and Norman Swan type websites. But I'm going to show you something which is a little more complicated. And it's basically the same drawing, but I've got a drawing showing you the messenger RNA coiled up inside the virus. And it's the messenger RNA which carries the information to make new virus. And you can see the red spikes on the outside. Of course, these are all totally false colours. Okay, so let's go back to uni and do our cell biology 101. And I've drawn up a very simple cell where you've got a cell membrane, the cell itself, the nucleus, and the cytoplasm. And we're going to be using these terms henceforth. Okay, if you ask me what I am, I could tell you I'm a molecular biologist. And therefore, I know something about the transition from DNA, you've all heard of DNA, to messenger RNA, which carries what DNA had, only it carries it on a single strand. And then that single strand goes to the cytoplasm and makes protein. So something to think about. SARS-CoV-2 is, if you get the infection, is interacting with your lung cells. So where on the cell do you think SARS will bind? Remember back to that membrane? The answer is it binds to the membrane. And which part of SARS binds to cells? You really do know this one. What's sticking out from the spikes? That's it. I think you sort of know what messenger RNA is by now. And you know what it does in the cell. You've all heard of the word nucleic acid. You've all heard of DNA. So your messenger RNA is the thing that codes for protein molecules. And of course, we're interested in what messenger RNA does when it gets into the cytoplasm. So I won't go into that. So for this talk, of course, the protein of interest is the spike. So here's a nice picture. I don't know whether I showed you this one before. It's an electron microscope picture of one cell, believe it or not, in the lung. And you can see one of our breathing tubes right in the centre. And you'll see bright green spots. I think, yep. That's the actual size of the virus compared with a cell. Okay, so what happens when you get a SARS-CoV-2 infection? The messenger RNA is going into the cytoplasm and it enslaves the whole cell. So the whole cell is converted to a factory for making new virus. And eventually, of course, that virus must emerge from the cell. And there's another nice electron microscope picture of a cell in blue showing new viruses coming out. And new virus travels freely around the body in the blood. So it can go and find any other cell and bind to it, and the infection can continue. And it's when this happens in a very strong way that that person is admitted to intensive care. So just a quick thing about how we develop immunity. Your immune system learns to recognize foreign invading proteins. That's the whole basis of vaccines. So those foreign proteins are called antigens, and you've all heard of the word antigen because you make use of it when you do a rapid antigen test. So your vaccine is inactivated antigens. In other words, just blobs of protein. So those antigens from the vaccine now enter your bloodstream when the nurse or the doctor gives you that injection. And this has to happen slowly, otherwise you get anaphylactic shock. And that's why the doctor or the nurse tells you to go and sit down for a while, for 10 minutes or 15 minutes, to make sure you don't get a bad reaction. Now, antigens, once they've come into your blood, cause the production of antibodies. And perhaps you might have sort of guessed that antibodies are our key immune defense. So as soon as SARS-CoV-2 gets into your system, some of the plasma cells, so-called plasma cells in your blood, immediately begin to make antibodies, which hereinafter I call AB. But then longer lasting cells, which are called B cells, found in the marrow of your leg bones, begin to make antibodies. And the antibodies made by the cells in your legs usually last for decades. So now we're going to be starting to look at the genetic variants of SARS-CoV-2. So, and each variant produces its own type of spike. So each spike protein of that new variant is therefore a unique antigen. And as we've just seen, each antigen tells your immune system to make specific antibodies to that antigen. So here I've listed the variant antigens that we know thus far. You've all heard of alpha, beta, gamma, and mostly you've heard of delta. That's what Pfizer used to make vaccines. Pfizer's vaccines are all against delta and Moderna and the rest. But in December, I think it's December last year, along comes Omicron. It has 32 new mutations, and 10 of them are in that area of the spike that binds to cells. So it's a tricky beast. And something I enjoy is telling you how to pronounce it. Variant is pronounced Omicron. You don't say, perhaps some of you would say omega, but you usually say omega, don't you? Because that means, omega means capital O. Omicron means little o. So it's not Omicron, as newsreaders tell you. So here are two nice pictures. You can guess that they're pictures of Africa, but in fact they're not. They are pictures of the new type of spike. The delta spike is on the right, and it only has 9 mutations. The Omicron spike is on the left, and it has 32 mutations, as I've just told you. So those are protein maps of the spikes. So, as a sort of aside, what do antibodies do? Well, they circulate in the blood for months to years and decades, and they are able to recognise newly invading antigen. Once they've recognised it, they lock onto it, so you get an antigen-antibody molecular complex. Now these things are in the blood and they are huge. But the activity of the antigen, by being complexed, is neutralised by the antibody. So here's some nice pictures that were developed by people at Monash University in Victoria. And they show antibodies in green approaching a virus. And they lock on to the spike. And here is the latest picture of that, where the little pink thing at the bottom is the spike, and the huge blue and greeny thing is the antibody. So you can see the difference in size. So as I've said, the antigen-antibody complex is a very large molecule, and the enzymes in the blood don't like large blobs of protein floating around, so they begin to destroy that complex. So that whole complex is destroyed, antigen and antibody, and the outcome, of course, is the disease that you had is cured. Now, you've probably realised that all viruses are pathogenic by definition. They cause disease. You've got your coronaviruses, you've got poliomyelitis virus, and that's newsworthy because the trendy millennial parents have decided that they don't really need to vaccinate their children, and an outbreak of polio has occurred in New York and in London. The morbillivirus causes measles, the adenovirus causes your common cold, the rhinovirus causes the dripping nose that you get when you have hay fever. And of course you're all familiar with the influenza A virus, which is commonly called the flu. And we know about the Ebola virus, sort of. So now let's delve into our vaccine. Of course, this is the pointy end of the lecture, for want of other humour. Okay, what type of vaccines can we make? Well, there are 4 of them. We can either make them against the whole virus, and there's a group in France who are doing this, and they're up to phase 3 human trials, and that was at May this year. You can use what's called a viral vector, which starts with the DNA, and that DNA is transferred into messenger RNA, and then that's the method of AstraZeneca at Oxford. You can have what's called nucleic acid vaccines, where people like Pfizer or Moderna put the spike mRNA directly into the vaccine. Or the 4th way is to make a preparation of pure spikes, which is not difficult to do. And then so all those spikes in their concentrated tube are going to make antibodies. And that's what Novavax, you've all heard of Novavax, so that's what Novavax is. So I'm going to do a little test. In the vaccines of AstraZeneca and Pfizer, what is happening to the messenger RNA? What did I tell you about messenger RNA? What does it do when it gets into the cytoplasm? Makes protein, right? So the eventual antigen then is spike. And so what immune process does the antigen stimulate? That was literally on the last slide. You are allowed to go to sleep, but I'm hoping you're not. So the immune process that the antigen stimulates is antibody production. Now, a fact you didn't know. None of you will know this, but the wise owl does. Okay, SARS-CoV-2 by itself can cause blood clots. So think back to when Mr. Multi-Ministries decided that he would listen to ATAGI. Remember ATAGI, the medical advice thing? And they said, oh, well, AstraZeneca is no good anymore, it causes blood clots. So anybody under 50, we're not going to recommend that they have AstraZeneca. So that was all a big lie. And here I've got an electron microscope picture of clumped red cells and the fibrin protein around them that makes the clot. Now, SARS-CoV-2 can make that happen just by itself. So here I've got a few take-home messages. If you can activate the spikes, then the virus can't get into your cells. So all of those all of those vaccines produce antibodies which inactivate spikes. So each of those 4 vaccines will prevent SARS-CoV-2 from getting into your cells. So if you're vaccinated and boosted twice, you can't succumb to major infection. You might get a minor dose, but it won't send you to hospital or kill you. So what's happening then? How did the Omicron virus, how did the Omicron variant come onto the scene? Well, because of their very nature, nucleic acids can suffer what we might call mutations. And these are translated into differences in the messenger RNA sequence. Some of these mutations, for whatever reason, perhaps a higher power, decides that they will be kept and replicated. And the latest spike protein variant is Omicron. And you might, if you're thinking about it, all of Pfizer's and AstraZeneca's and Moderna all used Delta as their reference point. So you all will have strong immunity against Delta spikes. So that's okay if you get Delta variant. But if you get Omicron variant, it might be able to evade some of your antibodies. So, as of December 2021, oh, you've seen that sort of data before, sorry. Now, Omicron is a very transmissible virus, and its numbers double every 2.5 days. So you can see that it is very adept at going through the population. But the Omicron variant is much less likely to send you to hospital or kill you. So let's go back to uni again, and we're going to be looking at the lower respiratory system. The upper respiratory system is from about Adam's apple upwards, in other words, nose and throat. You've got your trachea, which is your main breathing intake tube. You've then got your bronchi, left and right, which goes to the lungs. And you know about bronchi because some of you have had bronchitis in your lifetime. So here I've got a very simple diagram which is showing, it's comparing Omicron and Delta. And it's looking at what's in the bronchi and what's in the lungs themselves. And you might ask, well, what's it all about? It's showing you the virus multiplication rates in those two areas of your breathing system. So Omicron multiplies very quickly in the bronchi and very slowly in the lungs, whereas Delta does the opposite. It multiplies very slowly in the bronchi and quickly in the lungs. So we know about it evading antibodies, and the latest news, which I don't think has been released by the powers that be, is that Pfizer is now developing vaccines specifically made against Omicron. So we're trying to keep a step ahead. So here come some graphical representations which are easy to follow. As the vertical axis tells you about the frequency that Delta is found in the population, it also tells you about the frequency that Omicron, and you'll see that Delta gradually has been eliminated from the population, if you like. But, as I've said before, December 21, Omicron is suddenly on the scene and it starts to increase. And that data was from January the 3rd this year. So let's concentrate on Omicron's new identities. You can see there the early SARS variants, the Beta, the Delta, and then the Omicron. And those spikes, I shouldn't use the word spikes, those peaks occurred roughly every 6 months. So every 6 months, a new variant comes on the scene. And the latest variant to come on the scene, which is now dominant, this is data from May this year, is BA4 and BA5. I'll skip that. So Omicron's BA4 and BA5 are now dominant. There are almost 100,000 new cases per day across Australia. And the total Australian deaths from COVID disease exceeds 11,500 across Australia. And so you might be asking yourself, well, what good did the boosters do? Well, here's a beautiful demonstration of what they did. Before the booster, you can see all the different types of variants of Omicron, and they're relatively low antibody production. But as soon as you get the booster, the antibody production goes up enormously, as you see on the right-hand side of the graph. And remember, well, you won't know, but the vertical axis is what we call a log scale. So every division is 10 times the previous decision. So it goes 10, 100, 1,000, etc. Now here's a graph of hospital admissions for all of this year up to 2 weeks ago. And it is one of Norman Swan's graphs. And the yellow tells you how many thousand people are hospitalised. And the red tells you how many are in intensive care. So we could call those the 3 peaks of Omicron, which they are. So they're gradually getting better at preventing you from dying. And as I said, Omicron is very unlikely to kill you. In other words, it's very unlikely to send you to intensive care. Here's a slightly depressing graph, which shows you that at August the 12th, in other words, not too long ago, here we are, the cases per 100,000 versus the age band. So you'll see that the people who are in their 20s have the most number of cases. That's the third group down. Whereas we, who are maybe some of us are in our 70s, aren't doing too badly in cases. That wasn't put up to depressing. Here's data for Victoria, which is COVID-19 deaths per age band. And you'll see that in Victoria, deaths are worst in the 80 to 89 age group. Remember, this is deaths, this isn't cases. And here's another one of Norman Swan's slides. This is death across Australia across the year so far. And it doesn't require very much description to tell you that deaths are increasing as of August, despite Omicron being dominant. I can't explain that. Okay, you might be wondering whether you should be wearing masks. And this is showing you the effect of compulsory mask wearing, which is occurring in Singapore, Japan, and South Korea. We are much more laissez-faire in Australia and New Zealand. We think it's un-Australian to make people do things. So our deaths, as in the blue graph and the pink graph, are increasing because we are not wearing our masks properly. Whereas in Singapore, Japan, and South Korea, which used to be bad, the government said, right, you will wear masks. And their death rates have plummeted. So here's the SARS-CoV-2 virus on the atomic scale. So it looks a pretty nasty beast. In this case, those turquoise colour things are the spikes, bristling with with molecules to grab your cells. And I thought I should tell you about the Scottish variant from north of Hadrian's Wall, and at least you can hear it coming. Okay, that's the end of the talk. My time has run down, and Salvador Dali cleverly produced that picture. And now it is your turn. Yes, please, fire away. I'll show you my ignorance. Yeah.
You have two questions, Steve. When we have a PCR, does that tell the authorities which variant?
Yes, it does. And most PCRs will be against Delta. They are against DNA sequences that produce Delta.
What's a PCR?
What is PCR?
Yeah.
It is called polymerase chain reaction. It's a way of multiplying the little amount of DNA that you can get from a saliva sample, for instance, and they can multiply that DNA but still keep its characteristics. So that's what PCR is doing.
So again, why if we've got Omicron and the BA4 and 5, why are they not showing? Why is it still Delta showing the PCRs?
They haven't produced the parts of the PCR to multiply the DNA that produces BA4 and BA5. It's too far ahead.
So that would apply to the RAT test also? Is that exactly the same as it
No,
was?
the RAT test is different. The RAT test looks at the antigenic properties of the viral envelope. In other words, the RAT test isn't looking at spikes. It's giving you a much more definite indication because it's looking at the viral envelope. In other words, the green bit, which I'll get back to. It's a long way back. There it is. Now your RAT test looks at the green bit. It's looking for antibodies that you've made against the green bit. It's not worrying about your spikes. So in fact, when you think about it, every virus, every variant of this virus is going to have the green bit. So if you can make an antibody or whatever against the green bit, which the rat test does, then you can test for any type of SARS-CoV-2. Because you're not relying on there being changes in the spikes. So does that help any? Yeah. The PCR business is far too difficult to explain. I could, but then we'd need another hour. So I won't.
So, Tim, why
Right.
then, when you do the rat test, and then you have to go and do the PCR?
Oh, but you only have to do the PCR if the rat test is positive.
Yes.
In other words, the rat test is showing that you've got antibodies
All right.
to the green bit.
Oh, okay. And
So-
then the PCR proves it.
PCR proves it. It won't tell you what variant you've got. It'll tell you you've just got the virus. Yes.
Sorry, may I ask another question? Could you tell me the action of the antivirals, please?
Yes. The antivirals, they look at the messenger RNA and they prevent you from making more messenger RNA. Simple as that. Messenger RNA is made up of 4 different types of chemicals that are tacked together. And if you can, even just one of those chemicals, which is your antiviral, doesn't have the right structure, so it causes a major hiccup in the multiplication of messenger RNA. So that's one of the reasons why you have to get it pretty early after your first symptoms. But then it works. And that's the same with all antivirals. They attack either the DNA or the messenger RNA and just prevent the virus from multiplying. Yes?
I know you said it's another lecture, but are you able to give us some sort of brief description of what's going on in moving into is it post-acute viral syndrome, i.e. long COVID?
Yeah.
But other viruses do the same thing, the process.
So would you like
Tell us
me to...
briefly what's going on in that process, moving from the acute stage to that long-term stage?
All right. Well, if you can bear with me, I will go forward again, backward, to long COVID. Because I'd actually hoped that I didn't need to put a stooge in the audience to ask about long COVID. So you just have to bear with me. So you see, it takes a fair amount of application, if you like, to make a talk like this. I've got another talk about the latest developments in how to get spike into you to make antibodies. But here we are. So here are the long-term SARS-CoV-2 infection effects. They can affect cardiac muscle cells, in other words, the actual cells of your heart muscle. They can affect your brain cells, your kidney cells, and, not surprisingly, your lung cells. Okay, here is a picture of what happens when SARS attacks your cardiac, your heart muscle cells. The unaffected is on the left and the affected is on the right. So long COVID is that thing on the right. So you can see cells have been broken up, and let's go on. Here is the case in the brain, which is giving you what people call brain fog. So in your brain you have cells which are called astrocytes for some reason, surprise, and they can actually be reduced and destroyed by SARS-CoV-2. And here is an example of what's happening in brain fog, where you have your normal astrocyte with all its nice projections, if you like, and you have an affected cell where SARS has virtually killed off the nice long spiky bits that do all the correct communication. So, kidney effects. In the kidney, you have excess levels of protein in your urine due to SARS. You even have blood. And in general, SARS-CoV-2 is depriving oxygen supply to your kidney cells. And you know that how important the kidney is. Okay, lung effects. You know this too. Shortness of breath on exertion and asthma-like breathing difficulty. So you can get over that by using the asthma puffers. Looks like it.
Is it too early to tell how permanent those effects will be? Or can we recover from those effects?
I think the brain stuff you probably can't recover from, unless, although we do know that the brain is plastic, so it can make new cells. Mm-hmm. So it might, for some known reason, start making those spiky things again.
Right.
Kidney, I don't know how you'd recover from that.
Not everyone will get long COVID.
What was the everyone? Would everyone?
So some people will get it and some people won't get it.
Yes. Yeah. It just depends on which cells the damn virus attacks.
And how do we convince the governments of not tell us to wear masks?
I don't know.
You've got a graph there that shows what happens.
Yes.
And they know about those things.
But they don't. That never comes. That is never released to the press because it is too depressing.
Okay, well, can we thank Tim for all the information that he got today? Yes, that was a bit mind-blowing, wasn't it? A bit mind-blowing to hear all that information, but it's good to know and have that in the back of your mind when we're walking around, and I will definitely put my mask on more often in the next 2 weeks. Thanks very much, Tim, it was really fabulous.